Hello everybody, my name is Tasin Tarnum and today I'll be talking about the DNA structure using the Watson and Crick model of DNA structure. According to this model, DNA has a structure of double helix where two strands are antiparallel to each other, having 3' and 3' and 5' ends. The nitrogenous bases are adenine, guanine, cytosine, and thymine. These are complementary to each other and binds with each other through hydrogen bonds. A binds with D and C binds with G. Due to the helical structure, it has major and minor grooves, and in a stable DNA, the width of a helix is 2 nanometers and width of a complete turn of a helix is 3.4 nanometers that contains 10 base pairs. Let's talk about the components of DNA. DNA requires a 5-carbon sugar. These carbons are marked down from 1' to 5'. The 2' carbon plays a significant role in determining whether the sugar is for DNA or RNA, because if the 2' carbon is attached with hydrogen, it's the deoxyribose sugar for DNA, but if it is attached with OH, then it is the ribose sugar for RNA. This is the phosphate group, and the nitrogenous bases are adenine, guanine, which are purines with 2 ring structure, and thymine, cytosines, which are pyrimidines and with single ring structure. Uracil is for RNA. Here, the phosphate group is attached to the 5' carbon of the sugar, and nitrogenous base is attached to the 1' carbon of the sugar, and together they make a nucleotide. The phosphate group and the sugar is attached to each other through phosphodiester bond, and this is how the backbone or the phosphate backbone of DNA is formed. How do we determine if it's antiparallel? The 5' carbon of the sugar is facing upwards in this strand, and the 5' carbon sugar is facing downwards in this strand. So this is 5' to 3' strand, and this is 3' to 5' strand. Here, complementary nitrogenous bases A and T are attached to each other by 2 hydrogen bonds, and G and C are attached to each other through 3 hydrogen bonds. What is the stable structure of DNA? This is the stable structure of DNA because the nitrogenous bases are hydrophobic. So in this structure, the hydrophobic nitrogenous bases do not come close to the watery materials of cell, and the phosphate backbones are hydrophobic, so they like or prefer staying in this structure. If the phosphate backbones are close to each other, then it is the minor groove, but if it's farther apart from each other, then it is the major groove. The distance between two nitrogenous bases is 0.34 nanometers, and these are the distances that I have elaborated before. There are 2 types of DNA, the right-handed and left-handed. How do we determine this? If the closest phosphate backbone is moving up towards the right, it is the right-handed, but if it's moving up towards the left, then it is left-handed. In this picture, this DNA phosphate backbone is moving towards left, so this is DNA, left-handed DNA. This is also moving towards the left, so it is left-handed DNA. In this picture, we can clearly see this is moving towards the left, so it is left-handed DNA, and this is also moving towards the left. In this picture, this strand is also moving towards the left, so it is left-handed, and I also attached links, so thank you for watching.